Hi everybody, my name's Marcus Tindall and I'm Associate Professor of Mathematical Biology and Head of Department in the Department of Mathematics and Statistics at the University of Reading. Today what I'd like to talk to you about is the modelling of spatial protein localization within cells. The model system I'm going to be looking at is that within bacteria. I'm going to be looking at two species of bacteria in particular, Escherichia coli and Rhodobacter spheroides. And what I'm going to be demonstrating is the importance of how modelling those systems and where the proteins are localized does affect the outcomes of the actual processes you are investigating. So without further ado, what I'm going to do is start moving through my presentation. What I'll do is look first of all at examples within E. coli, and then I'm going to move on to the model system of Rhodobacter spheroides that we've spent a few years looking at here in collaboration with colleagues at the University of Oxford. What I'd like to start by doing is looking at the actual response of bacteria to stimulants in their environment and how they move, and then link that on to the physiology of the cells and respectively their biochemistry, and then how the stimulus affects the actual receptors on the surface of the cell with then their response to that environment. So what you can see here is cells, E. coli cells moving under the microscope and responding to different gradients of attractants in doing so, or the attractant gradient in the localized area around the cells. In a moment when I replay this, have a look at the cells in the center here. They are tumbling, some of them, and that is being used by the bacteria to reorientate themselves and go in a direction where they may be able to find attractant they've not been able to locate so far. Other cells are moving quite strongly in a very directed motion called chemotaxis, and we see here that the flagella on the surface of the cells, where in the previous case they were being flailing apart, actually come together in a very tight bundle. So this allows us to then look at the actual makeup of the E. coli cell, which is comprised of the following. It's a rod-shaped type cell with hemispheres at either end. It has a receptor array at either end as well, but I'm going to describe it here on the left-hand side. The biochemistry and the makeup of bacterial chemotaxis cells, in particular E. coli, has been studied quite a lot in the last 40-50 years, and quite a bit of detail has been elucidated, although that continues to be worked to this day. So first of all, we start with the key A protein. Here any key protein or CHE protein refers to a chemotaxis protein. Chemotaxis protein A autophosphorylates in the absence of any attractant gradient to pass phosphoryl groups to key Y and key B. Key Y thereby phosphorylates to become the phosphorylated version, and both these proteins serve important response regulation in terms of how the flagellar motor respond. So in the case where we have quite a lot of key Y phosphate, we have that the cells, the flagella, spin in a clockwise response, thereby the flagella flail apart, and you see the reorientation type tumbling behavior you saw in the previous slide. When we have reduced levels of key Y phosphate, in other words, just basically key Y, then what we see is the flagella actually come together in a counterclockwise rotation, and that allows the cells to then form these quite helical bundles you also saw in the previous slide. I'm going to be focusing mainly on these proteins here today, key A, key AP, key Y and key YP, that's the phosphorylated versions respectively of each of them, and also the phosphatase key Z, which acts to dephosphorylate phosphorylated key Y. There are these other proteins, key W, which anchors key A to the receptor cluster. Key R and key B both act in adaptation, although I'm not going to go into a lot of detail about them here today. So why would we want to study bacteria? What's our motivation for doing so? Well, bacteria, their biochemistry and their connection with their physiology is relatively straightforward, but it's also quite complex. There's a lot of other dynamics going on below the surface here of how these cells respond to their environment on the individual scale. Likewise, how does the individual scale behavior affect the overall population response? We've done work in that in previous years. And this leads us on to look at bacterial colonies, commonly known as biofilms. We remove them from our teeth each day, they foul up ships, and also they can be found on a range of other surfaces, for example implants and so on and so forth. So from a range of viewpoints, how can we use their understanding of bacteria from the single scale to the actual population scale to describe, manage and control them. Okay, so another thing from a modelling point of view that makes E. coli a very attractable system to model is the range of experimental data that has been collected, and quantitatively so, over the last 50 years. This allows a range of values for describing the reaction kinetics of the process as I described on the previous slides, as well as even diffusion coefficients have been elucidated in studying this system. That's less so the case a few years ago with other cellular systems, but more and more, and I know this is the case with this conference, as people begin to understand single cell phenomena, diffusion processes and so on and so forth, this data is becoming a lot more widely available for a range of single cell systems. And so the approaches I'm demonstrating here can be applied to lots of other cellular systems where relevant to the questions one may wish to have answered. What I'm going to be doing in the next few slides is looking at different mathematical approaches to modelling the systems of bacteria we're going to be looking at. We started here with E. coli. I'm going to move a little while after that to looking at Rhodobacter sphaeroides, another type of bacteria we've been studying. And I'm going to look at solving these systems numerically, that's via a computer. We're going to be using ordinary differential equations to look at Rhodobacter. You can get analytical solutions, but that means by pencil and paper only. But it's quite hard to do so because the systems here become quite large, complex, non-linear in terms of the interactions between the respective proteins in the system. So we're going to be looking at numerical simulations in a moment as well on MATLAB. Partial differential equations allow us to model the spatial effect, the spatial localization of the proteins and also the reactions between them, and incorporating diffusion, and in so doing we therefore model what is known as the spatio-temporal dynamics. This allows us to solve these systems of equations using software known as COMSOL is one example, and there are other ones available as well. Okay, so mathematically, I'll just briefly outline here what we're going to be looking at. If we look down here on the bottom right-hand corner, we have an abstract representation or a schematic representation of what a E. coli cell is going to look like. We have here that this omega-1 region is the cytoplasm of the cell, and we've constructed two receptor clusters, omega-2 and omega-3 in this way. We also have the surface of the cell to consider, where we don't allow, obviously, any proteins to move outside that region. We define here within the cytoplasm of the cell, remember we're just looking at key Y's and key A's to begin with. We will include key Z in a moment, and I'll tell you more about that. The key Y diffuses, likewise its phosphorylated form diffuses within the cytoplasm of the cell and dephosphorylates there. The key A's are restricted to the two receptor clusters at each pole, and they actually autophosphorylate, but also pass the phosphoryl groups to the key Y's. That's what the dynamics here on the right-hand side of these equations is demonstrating. The key Y's and their phosphorylation version diffuse also across the receptor clusters, allowing them to pick up the phosphoryl groups from the key A. But in the moment we're going to be considering cases, one of which is the fact that the phosphatase key Z can be contained at the receptor clusters only. So we put all this together and we simulate what happens within an actual E. coli cell. What we can see is how the key Y becomes phosphorylated through the reactions at the receptor poles and thereby diffuses within the cytoplasm of the cell. It eventually builds up. This is in the case where there is no attractant gradient present for the cells to respond to, and we can do other simulations where we have attractant being added to the cell surface or removed and so on and so forth. Here we're just looking at the case where there is no attractant present. So in doing so, we can now use our model to begin to postulate and understand what other things may be affecting the response of the actual cells to their environment. Important is the concentration of key Y and key Y phosphate within the cytoplasm of the cell. What we want to ensure is that it's relatively constant. There's not a huge gradient difference within the regions because of the flagella located on the surface of the cell. If we have lots of flagella on the surface of the cell and we vary that concentration gradient a lot, then there will be differences in how the cell responds to the environment, and we won't be able to get those nice tight bundles of flagella forming, which allows bacterial chemotaxis to occur. So this little experiment here in terms of computational represents what we can also do in the lab to some extent, although a little bit more difficult to do, is in here we've looked at here with the red dotted line the fact that key Z, the phosphatase, is only at the receptors of the cell, but the blue solid line is when it is within the cytoplasm of the cell. And we can clearly see that when key Z is at the cell receptors or localized towards the cell receptors, it's more likely to lead to a less decreasing gradient in phosphorylated key Y occurring, and it's more likely to be the case. Although we can't exactly determine this precisely, we can look at this in a bit more detail numerically, as we've done so here. So again, further added arguments for this is the fact that when you have a key A here, this point A here and this point B within the cytoplasm of the membrane of the surface of the cell where the motors would be located, you obviously don't want to have huge difference between them. And what we find here is that when we look at point A, when we have key Z at the receptors of the cell, or likewise in the cytoplasm, then actually the difference is less when we have key Z actually at the receptors here. So here, where we are looking at the fact that we're agreeing here in the sense of the red dotted line and the blue circled line, then we see there's very, very close agreement, whereas in other cases they will differ in that way. Moving on, what we can do is we can extend our model to include other things. We have looked, as in previous work and an associated work, at the localization of key Y to the receptor poles. I'll look into that in just a moment and demonstrate the importance of that. But the key B work and also a simple model of adaptation, whilst we have included that in other related work I'll talk about with Rhodobacter, I won't be talking about that in more detail today. So, although we can still have detail here in this slide demonstrating how key A becomes phosphorylated and key Y phosphorylated becomes dephosphorylated by key Z, and likewise phosphorylated key Y just naturally returns back to key Y in its unphosphorylated form, there are a lot more intermediary complex reactions occurring between a phosphorylated form of key A and key Y itself to actually phosphorylate key Y. And this involves not only phosphorylated key A, but unphosphorylated key A in that sense. And we can see here there's a range of complexes formed in the intermediary steps before we reach phosphorylated key Y here. So what effect does that have on the distribution of key Y and key Y's phosphate within the actual cell itself? Well, what you are seeing here is you're seeing slices of time-simulated responses of a cell to varying cases of key Y, where the key Y is located at the actual polar clusters in the way in which we have described in the previous reactions. So here you'll see that this is free key Y, also key Y bound at each of its complex forms with phosphorylated and unphosphorylated key A. And what this leads to is it leads to demonstrating that key Y can become localized to the receptor cells, receptor clusters, and also that it is less likely to occur within the cytoplasm of the cell. And this actually, in this work we did a few years ago, demonstrated that the cells themselves in this way, in terms of experimental data, is a lot closer in agreement than when actually you just have lots of key Y floating throughout the cytoplasm of the cell. So this extends your model, it includes more detail, and that comes about by understanding the biochemistry through experimentation as well. So the two go hand in here as we move forward together. Moving on to Rhodobacter spheroides, which is a water-bound bacterial species, we are interested here in understanding how the physiological response is tied again to the biochemistry of the cell. So here what I've again looking at is receptor clusters. This is a rod-shaped bacteria with hemispherical ends, receptor clusters at either end, but in this case there's also a cytoplasmic cluster here in the center. And the biochemistry underlying this system is somewhat more complex. There are a range of proteins here which have a range of processes which are not all yet fully been elucidated in a number of ways. So to begin with, the kinase KA2 is only located at the poles of the cell, and it becomes autophosphorylated there itself in the absence of any attractant gradient. I should also point out that Rhodobacter cells only have actually one flagella. So when they rotate their flagella, they can actually propel themselves through the environment in response to attractants themselves. When they don't move, in response to an attractant, they just sit in the environment and are buffeted around by molecules within the environment itself. So coming back to the biochemistry, we have key A2 located at the receptor poles. Key A3 is located only at the cytoplasmic cluster. And notice here the reactions are very different. So whilst phosphorylated form of key A2 drives and phosphorylates key Y3, key Y4 and key Y6, all of which are needed to drive the motor response, key A3 here in the cytoplasm of the cell is only responsible for driving and phosphorylating key Y6. Likewise, I've included key B here, and we'll have a look at all of these in a moment in a bit more detail, specifically looking at the motor driving proteins key Y, 3, 4, and 6. But the key Bs here are also phosphorylated in a different form. Key A2, in its phosphorylated form, does both of them, but key A3 only does key B2. And there's a lot more work being done as to understand why this is the case. So the question then comes about in this work as to how the two clusters may in a way be related, given there's all these reactions going on. Given that in this case I'm going to be modelling the system using ordinary differential equations, we can look at basically the reaction kinetics between the two systems without the necessary need or complexity for adding in diffusion. We're not particularly interested here in understanding the spatial temporal profile of the actual proteins. What we're interested here is in how we connect the two clusters possibly together through their dynamics. So we're just assuming that when the proteins are being phosphorylated, they're able to diffuse around. Obviously, some are located only at the cytoplasmic and polar clusters, those being the key A3 and the key A2 respectively. So there's a lot more work that needs to be done in a system like this to understand how the proteins move around and also how much spatial localization they do have. In terms of informing this system, a lot of data was collected, and this data was fitted during this work to understand the reaction rate constants that for other systems like we saw with E. coli are so readily available within the literature, but less so the case here for Rhodobacter. So in fitting the dynamics, what we find is that this allows us to then determine the reaction rate constants, which in this case here we have scaled with respect to how rapidly key Y6 in its phosphorylated version dephosphorylates, what the X's mean here at the bottom of the screen or the diagram. And in fact, we see some reactions are happening very, very quickly, in this case of Y4 becoming phosphorylated and Y6 becoming phosphorylated, each in terms of what's happening at the receptor cluster by key A2 action only or key A3 respectively. And likewise, we see some reactions are happening a lot slower in that way as well. What we can do is we could run some experiments here, numerical experiments, computation experiments, to understand what may be any connection between the system itself. So we run an experiment where we turn off the cytoplasmic cluster processes. We find that A2 autophosphorylates. It transfers to phosphoryl groups to Y3, 4, and 6, giving the response regulator protein levels of 3, 4, and 6 in their phosphorylated forms. We can then also do the experiment where we turn off any processes occurring in the receptor clusters and see what effect any key A3 phosphorylation has upon any other parts of the system. Now, one would expect if we turn off these processes to the left that we would only see phosphorylated versions of key Y6. Actually, what we saw in our simulations was the fact that we got phosphorylated versions of all of the proteins, the motor driving proteins 3, 4, and 6. And here what we've done is we've just simulated the effect of a ligand being applied. In other words, those phosphorylated forms would decrease, and thereby the autophosphorylation process has been switched off. When we actually take the ligand off, then they become phosphorylated again and pass those phosphoryl groups to the motor driving proteins, the KYs. So this raises the question as to what's happening here. And going back and using these results to understand the system, we found that actually just by stimulating this pathway here on the right-hand side, the cytoplasmic cluster, you actually, by Y6 becoming phosphorylated, in the reverse reaction process can actually pass phosphoryl groups back to A2 such that it becomes phosphorylated, and so Y3 and 4 both become phosphorylated in that sense, hence the reason why you see their values decreasing when you add a ligand on in that sense. So this allows us to say that the two clusters are connected theoretically. What happens when you do experiments where you find actually it's not via the key Y's that this is happening, in particular key Y6, but it's actually via the key B's, and in particular key B2. So this was an interesting finding. This demonstrates that modelling has elucidated one mechanism. It's not exactly what you see experimentally, but it's led you down the path of investigating further, and it therefore allows you to further inform the system as to what's happening here in terms of its reaction kinetics and also how it's responding. I'm going to finish there for now. This work was undertaken a few years ago, but is still highly relevant today. In particular, a lot of work that we've done in modelling over the years has been spatio-temporal. We've done less so spatio-temporal recently because, again, we're looking at new systems where it's been mainly more temporal dynamics, in other words, the reaction kinetics, but adding space in once we know the details and the available data from experiments, then that's available to inform the models, and we can then go ahead and do spatio-temporal modelling that I've demonstrated here today. This work was undertaken with Stephen Porter at the time, who's now in Exeter. George Wadhams are now still at Oxford. Judy Armitage is recently retired from Oxford, and Philip Maney is still there. There's a few publications, and what I'd like to do is finish by closing and saying thank you to the BBSRC for funding this work, and looking forward to answer any questions people may have. Thank you.